This is lecture 18, and we're going to continue our talks about agglutination and precipitation. In this lecture, we'll be going over the different agglutination tests, including passive agglutination, reverse passive agglutination, coagglutination, and the agglutination inhibition test. We'll also be taking a brief look at some precipitation testing methods, including turbidimetry, nephelometry, and some passive immunodiffusion techniques, including the Ouchterlony double diffusion. We're going to begin with some agglutination testing, and the first one in this instance would be passive agglutination. There are other names for this type of test, including indirect or particle agglutination, but for testing purposes, we'll refer to it as passive agglutination. And this test allows us to put antigens on a carrier particle to help us see the reactions, to help the visibility of these reactions. And we can use a variety of different particles, including latex beads, synthetic beads, RBCs, silicates, etc. So we would put antigens on these carrier particles, let's say latex beads, okay? And if you can imagine, just the antigen by itself is really super small, so we would need a lot of agglutination in order to see that reaction. But if we put it on a bigger particle and line this particle with antigens around the outside of it, then we'll be able to see this reaction more clear, better. We'll have better visibility of this agglutination going on. So in passive latex agglutination, we'll be using latex beads coated with our known antigen. Okay, so then we are wanting to determine if the patient's serum has antibodies to this known antigen. So we will basically mix our serum with the latex bead coated antigen, and if we have agglutination, then we have a positive test, and then we will know that this patient has the antibodies to the specific antigen we are testing for. If the test is negative, then we'll have no agglutination, and this particular patient will not have the antibodies that we're looking for. What can we test for using this passive agglutination test? Well, one thing we can usually test for is the rheumatoid factor. So rheumatoid factor is very important to understand and to know, especially for other testing, because if a patient has an autoantibody such as anti-IgM, then we need to know if it's going to cross-react with IgG antibodies, etc. So one way we could find out if this person has this rheumatoid factor is to do a passive agglutination test. And we can put a latex bead coated with IgG and then add the patient's serum and to see if we have any agglutination going on. If so, then we can say that this patient does have an autoantibody to the IgG, and therefore more testing would be involved to determine what is causing this rheumatoid factor, this autoantibody. When a rheumatoid factor test comes up positive, typically this is associated obviously with an autoimmune disease. It can be rheumatoid arthritis or some type of other autoimmune disorder. This test is very easy to perform. It is what we consider to be a screening test. So when we're testing for the rheumatoid factor, like I said previously in the other slide, we have a latex bead coated with IgG, and this is our known antigen. and then we'll add the patient's serum because this is our unknown antibody. We don't know if they have antibodies to IgG or not. And then we'll mix the two together and get our test results, whether it's positive or negative. So here is the steps in the passive agglutination of what goes on. So remember, we start out with a carrier particle. It could be a latex bead, an RBC, synthetic beads, etc. Then we want to add our known antigen, and then this known antigen will bind with the carrier particle. So then we'll have our coated particle. Then we'll add the patient serum, alright, which are the antibodies that we are wanting to test for. And then if the patient does have antibodies to this particular antigen, then we'll see a visible agglutination. Now there are test kits. Typically these come in test kits, and the soluble antigen and the carrier particle are already mated together. So this is one step that typically techs do not have to do. So basically I'll just add the patient serum and look for the reaction. The next one is passive hemagglutination. So remember in the last lecture I said, remember if we ever have heme in front of agglutination, then we need to think of RBCs. Alright, so in passive hemagglutination, the carrier particles are RBCs. Typically we'll use this test for rubella. So it's exactly the same. We'll have rubella antigen coated on RBCs, and then we'll mix the patient serum because we want to know if they have antibodies to the rubella antigen. And then if we have agglutination, then we could say that the test is positive and the patient does have antibodies to rubella. The next test is reverse passive agglutination. So in this test, we are actually putting antibodies on the carrier particles. Obviously, we can use RBCs, latex beads, synthetic beads, etc. as these particles. So we will be actually putting antibodies on the carrier and detecting the antigen from the patient specimen. So we match mix the antibody carrier particle and the patient serum together, and if we have agglutination, then this patient specimen does have the antigen that we are looking for in their serum. Now you may think, okay, well, what's the difference between the rheumatoid factor latex coated antibodies and this? So in the passive latex agglutination, when we're testing for rheumatoid factor, the antibodies on the latex is an antigen. We don't consider that as an antibody because we're looking for that autoantibody IgM to the IgG antigen. So that is the difference between passive latex agglutination when we're testing for rheumatoid factor and reverse passive. So we're actually putting antibodies on the carrier and looking for an antigen in the patient serum. One of the main reasons why we would use reverse passive agglutination is to detect bacterial or fungal antigens within body fluids. Cryptococcus neoformans, for example, in CSF, we'll use reverse passive agglutination. So we'll have antibodies that are specific for crypto, and then we'll put them on the latex particle, and then we'll mix the CSF and the latex bead covered in antibodies specific for crypto, and then obviously if it agglutinates, then this would be a positive test and the patient would be positive for crypto neoformans. One issue with doing reverse passive agglutination is rheumatoid factor. If a patient has rheumatoid factor, then obviously it's going to bind, this autoantibody IgM is going to bind to these IgG particles on this latex bead when we're doing reverse passive agglutination. So it's very possible that we will get a false positive reaction if a patient has rheumatoid factor and we're testing for, let's say, crypto. So this is something that we need to look at, obviously, very closely, and doing a rheumatoid factor test first typically will help us know, whether or not this is a false positive or not. So again, here's a diagram of reverse passive agglutination. A lot of kits already come ready with the coated particle already with antibodies on there, so basically all we have to do in the lab is add our patient serum to our coated particles and then mix and look for agglutination. And of course, we can't have reverse passive agglutination without reverse passive hemagglutination. So, very same principle, except the carrier particle is an RBC. Typically, we'll use this hemagglutination to detect hepatitis B surface antigen in patients. The next test is coagglutination. So in coagglutination, the carrier particle is a bacteria cell, and typically this would be Staph aureus is used most frequently because it has what we have found to be protein A in the cell wall, which naturally absorbs the FC portion of IgG. So if you could try to picture that in your head, that we have a bacterial cell with antibodies sticking out around the surface of this bacteria cell. So the FC portion is dug into the cell wall, and its fab portions, the Y part, is still sticking out and able to identify antigens. So in this type of test, we know that the antibody is known because we have our known IgG that has been absorbed in the cell wall of the Staph aureus, and we're looking for an unknown antigen in the patient's serum. So in coagglutination, the antibody is known and the antigen is unknown, and the unknown obviously we will find in the patient specimen. So if we have agglutination and coagglutination, then the patient would be positive for the antigen we are searching for. If we do not have agglutination, then obviously the patient specimen does not have the antigen we're looking for. And again, here's a diagram of coagglutination. The next test is agglutination inhibition. So one thing you will definitely need to remember is the lack of agglutination is an indicator of a positive reaction. Okay, do not let this one beat you out on the exam. Okay, so the lack of agglutination is an indicator of a positive reaction. Please get that into your memory and go ahead and remember it for the rest of the semester. So agglutination inhibition is based on competition between particles and soluble antigens, or particulate and soluble antigens, for a limited number of antibody sites. And we'll go a little bit deeper into this testing in the next couple of slides. We perform agglutination inhibition pretty much every day in most labs. Maybe we just didn't understand what this testing was called, but pretty much all labs do pregnancy testing. So this is an agglutination inhibition test. So we have our patient specimen. Remember, we can use urine or serum. And we have our unknown antigen, which is our patient specimen. We have our known free antibody to beta-HCG, and we have a carrier particle with beta-HCG, which is our known antigen. So in the next couple of slides, we'll go through a positive test and a negative test and see how they differ. So let's walk through this testing. So we have our anti-beta-HCG, all right, plus we're going to add our patient sample. We're going to incubate, and obviously the antigen will not be present. In this case, the patient will not have beta-HCG. So then we're going to add antigen-coated latex particles. And so this anti-beta-HCG will then bind to these antigen-coated particles, and then we'll have agglutination, and this will mean a negative test. Okay, now let's look at the other side of this test. So we have our anti-beta-HCG antibodies, then we'll add our patient serum. In this case, we do have antigen or beta-HCG. Okay, so we will incubate those two together. The anti-beta-HCG will bind to the beta-HCG antigens. We'll then add the antigen-coated latex particles, and obviously they will not be able, the antibodies will not be able to bind to these antigen-coated latex particles with beta-HCG because they have already bound to the patient's beta-HCG, and therefore we will have no agglutination and a positive test. So these tests are quite easily performed. Remember, the interpretation must be carefully perceived, definitely because of agglutination inhibition, and a positive result is a negative agglutination. So that's something you want to be careful with. Remember, when during testing for my exams, typically I will give you QC results. Pay attention to those QC results. If the QC results are invalid, then that means the test is invalid. If controls or QC is not given, then assume the results are okay. I have highlighted some typical false positive, false negative reactions in agglutination testing, presence of cross-reactivity, presence of rheumatoid factor typically will give you a false positive reaction, and even a delay in reading the slide could possibly give you a false positive. Usually a false negative is due to the prozone phenomenon, which is, remember, we have too much antibody for the test. So look these over and be aware of especially the ones that are boxed out for you. Let's do a quick review and go ahead and answer these, and we'll go over the answers in the next slide. So a known antibody carrier with a test analyte and an unknown antigen. This would be agglutination inhibition. Antibody on latex and an antigen unknown. This would be reverse passive agglutination. Antibody on bacteria and an antigen unknown. This would be coagglutination. Identification of antibody or antigen. That would be direct agglutination. And antigen on latex and antibody unknown. This would be passive agglutination. So now let's take a look at precipitation. So what is the difference between precipitation and agglutination? So agglutination is the visible clumping of antigen and antibody complexes. Alright. So in precipitation, we actually have soluble antigens that combine with soluble antibodies to produce insoluble complexes. So, for instance, typically we do precipitation testing in agar, right? So we'll have our antibody or antigen in the center well, then we'll have our patient sample, and we'll allow those to diffuse out into the media. And where these antibodies and antigens interact, we'll get a ring of where they form these complexes. And this is what we are looking for in precipitation reactions. So the precipitation curve, just like the agglutination curve, has the same properties. So we want affinity and avidity in precipitation reactions, and we also want our antigens and antibodies to have multiple binding sites so they can interconnect and show us this ring of precipitation that we are looking for. And obviously we want these antigen and antibodies to be in relative proportions to each other because we want to get that zone of equivalence. So the zone of equivalence also plays a role in the precipitation reactions. So again, the zone of equivalence plays a major role in precipitation. If we have too much antigen or too much antibody, then we will not get the correct result in our testing. One other way we can measure precipitation is through light scattering. So we can use auger, or we could use this light scattering method. And when antibodies and antigens interact in a solution, typically that solution will become cloudy or turbid. And this initial turbidity is followed by precipitation, and we can use two different methods to detect this turbidity. We can use turbidimetry or nephelometry, and we'll talk about each in the next couple of slides. First, let's talk about turbidimetry. So this is the measurement of how turbid the solution is, or how cloudy this solution is. So light is passed through this antibody and antigen solution, and a detection device is placed directly in the incident light, in direct line with the incident light, and this device will measure the decrease in light intensity. Obviously, you do want to compare this to a blank, so this blank would be a QC, and then you would have your patient sample and measure it against the blank. Next we have nephelometry. So nephelometry measures the light scattered at an angle. So the amount of light scattered is directly proportionate to the concentration of the solution. Nephelometry is more sensitive than turbidimetry, and we can detect antibodies or antigens, and a lot of applications include we test for complement components, rheumatoid factor, antibodies, and other serum proteins, including acute phase reactants. Here is a drawing of the difference between turbidimetry and nephelometry. So in turbidimetry, we have the detection device parallel to the light source, and in nephelometry, remember, we're measuring the light scatter at an angle, so it is going to be put up at the angle to measure that angle light scatter. When we talk about precipitation testing, particularly in a gel media or agar, we are talking about passive immunodiffusion. So when we think about gel, we want to think about passive immunodiffusion techniques. Alright. Couple of things to take away from this precipitation method, especially in this passive immunodiffusion techniques, is that depending on the gel medium, how thick it is, how light it is, will determine the rate of diffusion. The rate of diffusion can be affected by particle size, the temperature, like I said previously, gel viscosity, and the amount of hydration in the gel. So these are all things that we have to consider when we are testing precipitation testing through passive immunodiffusion techniques. Now, using these techniques, we do not use electrical current to speed up the process. We just allow it to sit there and go at its own pace. The first passive immunodiffusion technique I want to talk about is the Ouchterlony double diffusion. It is called double diffusion because both the antigen and antibodies diffuse into the media. So we have our agar plate, we have our well that contains our antigen and a well that contains our antibody. Then we're going to let that sit and allow the antibody and antigens to diffuse into the agar. And where they meet is the line, what we call the line of precipitation. So this is where those insoluble complexes form, and this is also considered the zone of optimal precipitation. So if you think about the zone of equivalence, we have our excess antigen on one side, excess antibody on the other side, but in the middle we have our zone of equivalence. In the Ouchterlony double diffusion, we can detect antibodies or antigens. More commonly, we will try to be detecting antigens. So what is the procedure of this Ouchterlony double diffusion? So we have a gel plate with one central well and 6 outer wells. And I'll show you a picture of this on the next slide. Typically, we will put the antibody in the central well, so that means we are detecting antigens. And then we have our different unknown antigens on the outer wells with controls on the outer wells as well, mixed in with our patient sample. Then we're going to incubate. And we'll look at the next slide and I'll kind of talk you through it. So here's a picture of the Ouchterlony double diffusion. So we have our 6 outer wells and our center well. So in this instance, we are trying to detect the unknown antigen. So we have our known antibody in the center well. We have our 2 antigen controls, our known antigens, and then our 4 patient samples of the unknown antigen. So then we're going to put our known antibodies in the well, our known antigen controls in the well in 1 and 4, and then we're going to add our unknown antigen, which is our patient samples in wells 2, 3, and 6, and 5. We're going to incubate, and then after incubation, we're going to pull it out, and this is the results that we will see in this particular example. So our antibodies and antigens have diffused out into the media, and then we have this nice ring of precipitation. So in this instance, in this example, this would be a positive test. So the patients do have this antigen that we are looking for. As simple as it sounds, we do have different patterns that we can see. So we have 3 different patterns actually. We have the arc of identity, we have cross lines or non-identity, and then we have partial identity, which creates like a spur formation. So in the identity or arc of identity, the 2 antigens are the same. Non-identity, we have cross lines, the 2 antigens are completely different. And then we have a partial identity, which creates this spur formation, and that means that the 2 antigens are not the same, but they are similar. So typically we'll use this, I've seen this before in fungal IDs and even autoimmune diseases. So let's take a look at each one of these patterns individually. So the arc of identity. So this is another example of Ouchterlony double diffusion. Instead of 6 wells, we have 2 wells. Okay, so don't let that throw you off. So we have our known antibody. In this case, we are testing for Histoplasma capsulatum. So we have our anti-histo antibody in the center well. We have our QC, which is our known histoantigen. Okay, and then we have our patient sample, our unknown mold antigen. We are suspecting histo. So we're going to test it through the Ouchterlony double diffusion. So we're going to add our antibody, our QC antigen, and our unknown antigen, incubate, and then pull it out, and this is the result that we get. So this is what we consider the arc of identity. This is a positive test. So the unknown antigen is positive for antihisto. So that means this patient does have histoplasma capsulatum. So it forms this nice little arc, and this would be considered a positive test. So here we have our cross lines pattern, or our non-identity. So here we have our antibodies. We have antihisto and anti-coccidioides. We have our QC, which we have our known histoantigen and our known coccidioides antigen. So we're going to incubate this and let it incubate anywhere from 12 to 48 hours in a moist environment. Then we'll pull it out, and this would be the results that we would get. So our antihisto antibody is obviously going to bind to our known histoantigen, and our known coccidioides antibody will bind to our known coccidioides antigen. Now, we get these cross lines because the antibody to histo, is diffusing out and creating that red line all the way across because the antibodies are diffusing out that far. We're not going to have that arc of identity because the coccidioides antigen is not going to bind with the histo antibody, right? Because they're different antigens. So, but the anti-coccidioides antibody is going to bind with the coccidioides antigen and creating these cross lines effect. So we know that the antigen in well 2 is not the same as well 1, what we are trying to detect. So this is what causes this cross lines pattern, this non-identity. The next test pattern, or the last test pattern, is the spur formation. So this would consider a partial identity. So we have our antihisto antibody in the center well, QC histoantigen in well 1, and in well 1A we have our unknown antigen. So we'll add those in, incubate, and pull it out, and this would be our result. So why is this like this? So we know that our QC, our known histoantigen, will bind to the antibodies for sure, and that's what that red line is. However, our unknown antigen is very similar to the histoantigen. Maybe it has like one or two same epitopes that this histoantigen has, and this is what creates this spur formation. The antigens are similar, but they are not exactly the same, and this histoantibody will actually diffuse out further and connect with those more specific histoantigens further down the line, and this is what creates that little extra line past the unknown antigen line, that little spur, and this is what is considered partial identity. And here's another example of the actual real deal of what you typically might see in the laboratory. We have, well, in example number 1, we have our arc of identity. Number 2, non-identity. And 3 would be the spur formation. And that is all for Lecture 18. We will continue in Lecture 19 and wrap up precipitation testing.